everyone, welcome back to our channel Mbbs Artist. In this video, we will be learning about metabolism of carbohydrates. Let's get into the video. Whenever we hear rice, sweet potato, wheat, we think of carbohydrates. We are all fond of eating. Have you ever wondered before how these carbohydrates get metabolized in our body? Before that, let us know what is carbohydrates. It is a polyhydroxy compounds that contain carbonyl group, either aldehyde group or keto group. 80% of human caloric intake is of carbohydrates. Its functional properties are sweetness, chemical reactivity, and polymer functionally. Carbohydrates are classified as monosaccharides, oligosaccharides, and polysaccharides. Carbohydrate metabolism is a fundamental biochemical process that ensures the constant supply of energy in living cells. Carbohydrate metabolism occurs in three main stages: glycolysis, Krebs cycle, and electron transport chain. In this video, we will be learning about glycolysis. Glycolysis or EMP pathway is the oxidation of glucose to pyruvate and lactate. It is a sequence of reactions converting glucose to pyruvate or lactate with the production of ATP. It can be explained in three phases: energy investment phase, splitting phase, and energy degeneration phase. The first phase is energy investment phase, and the first reaction is glucose is phosphorylated to glucose 6-phosphate by hexokinase or glucokinase. Both are isoenzymes. This is an irreversible reaction depending on ATP and magnesium. The enzyme hexokinase is present in almost all the tissues. It catalyzes the phosphorylation of various hexoses like fructose, mannose, etc. It has low Km value for substrates, about 0.1 millimolarity, and is inhibited by glucose 6-phosphate. Glucose 6-phosphate is impermeable to the cell membrane. It is a central molecule with a variety of metabolic fates like glycolysis, glycogenesis, gluconeogenesis, and pentose phosphate pathway. And the second reaction is glucose 6-phosphate undergoes isomerization to give fructose 6-phosphate in the presence of the enzyme phosphohexoisomerase and magnesium. Third reaction is fructose 6-phosphate is phosphorylated to fructose 1,6 bisphosphate by phosphofructokinase. This is an irreversible and a regulatory step in glycolysis. That's about energy investment phase, and the next phase is splitting phase. Fourth reaction is the 6-carbon fructose 1,6 bisphosphate is split, hence the name glycolysis, to 3-carbon compounds glyceraldehyde 3-phosphate and dihydroxyacetone phosphate by the enzyme aldolase, that is fructose 1,6 bisphosphate aldolase. Fifth reaction is the enzyme phosphotriose isomerase catalyzes the irreversible interconvention of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Thus, two molecules of glyceraldehyde and 3-phosphate are obtained from one molecule of glucose. Energy generation phase. Sixth reaction is glyceraldehyde 3-phosphate dehydrogenase converts glyceraldehyde 3-phosphate to 1,3 bisphosphoglycerate. This step is important as it is involved in the formation of NADH plus H plus and high energy compound 1,3 bisphosphoglycerate iodoacetate arsenate. Inhibit the enzyme glyceraldehyde 3-phosphodehydrogenase. In aerobic condition, NADH passes through the electron transport chain and 6 ATP are synthesized by oxidative phosphorylation. The seventh reaction, the enzyme phosphoglycerate kinase acts on 1,3 bisphosphoglycerate, resulting in the synthesis of ATP and formation of 3-phosphoglycerate. This step is a good example of substrate level phosphorylation, since ATP is synthesized from the substrate without the involvement of electron transport chain. Phosphoglycerate kinase reaction is reversible, a rare example among the kinase reaction. The eighth reaction is 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase. This is an isomerization reaction. Ninth reaction, the high energy compound phosphoenol pyruvate is generated from the 2-phosphoglycerate by the enzyme enolase. This enzyme requires Mg2 plus or Mn2 plus as it is inhibited by the fluoride. The final 10th reaction, the enzyme pyruvate kinase catalyzes the transfer of high energy phosphate from phosphoenol pyruvate to ADP, leading to the formation of ATP. This step is also a substrate level phosphorylation and this reaction is irreversible. The products we get are 2-ATP, 2-NADP and 2-pyruvate. Glycolysis takes place in all cells. Enzymes are present in cytosomal fractions of the cells. It is a major pathway for ATP synthesis in tissues lacking mitochondria. It is very essential for brain, which is dependent on glucose. Glycolysis is a central metabolic pathway, and the intermediates provide branch to other pathways like synthesis of amino acids and fats. Reversal of glycolysis with alternative arrangement for irreversible reactions is called gluconeogenesis. Glycolysis under aerobic conditions gives pyruvate as end product, and under anaerobic conditions gives lactate as end product. Glycolysis is regulated by three enzymes, namely hexokinase or glucokinase, phosphofructokinase and pyruvate kinase, catalyzing the irreversible reactions regulating glycolysis. Phosphofructokinase is the most important regulatory enzyme in glycolysis. This enzyme catalyzes the rate-limiting committed step. It is an allosteric enzyme regulated by allosteric effectors. ATP, citrate and H+ ions are the most important allosteric inhibitors, whereas fructose 2,6 bisphosphate, ADP, AMP and phosphate are the allosteric activators. The inhibition of glycolysis by oxygen is known as faster effect. The phenomenon of inhibition of oxygen consumption by addition of glucose to tissues having high aerobic glycolysis is known as Crabtree effect. Basically, this is opposite to that of the Pasteur effect. The end product pyruvate is most important for TCA cycle. The pyruvate is further broken down into ATP, that is called biological coin. Hope you guys understand glycolysis. For further updates, subscribe MBBS at Ease. Thank you all.